All right, I'll take a look at number 1. Okay, so I'm looking at question 1. Now remember, we've seen there's maybe 4 different types of acid-base problems that we've learned how to solve. Refresh your memory of question 1. Which type of problem is question 1?
It's weak acid and conjugate base.
And why do you say that?
Because I get proton, or I get an well, I get a proton out of it and a negatively charged version of it, or the conjugate base of it.
Okay, that actually turns out to be a misunderstanding of what I meant by the different types. So those different types of problems are based not on what you get, but on what you start with. What did we start with in this problem?
We started with butyric acid.
That's right, but which of how would you describe that in more general terms?
It's not HCl, HBr, HI, or
so what I meant was that we want to use the types of different types of problems from that handout that I sent you. It's just that the handout describes what you start with, not what you end up with.
Mm-hmm.
So which of those types would this problem be based on what we started with?
It would be a weak acid and conjugate base.
Now, I'm not sure why you said that, because what my point is that the types are based on what you started with. So what did we start with in this problem?
Just a weak acid.
Yeah. Did we start with the conjugate base?
No.
No. The reason I'm emphasizing that is, how would you solve a problem where you started with both the weak acid and the conjugate base?
What you would do before, during, after?
Actually, that's a mistake. No, that's not correct. So
Is it that we do the Hasselbalch equation?
that's right. Yes. So I just want to make sure we're not confusing the different types of questions. So this problem, so you basically solved this question correctly. You did solve this question correctly. I just want to make sure we're understanding the method so that you can adapt what you might see on the test. So to review, what type of question is this?
Just a weak acid problem.
Yes, because those names of the types of problems are based on what you start with. So the method you used here is correct. You start with the start, change, end table. You don't use the Henderson-Hasselbalch equation here. Do you see that it wouldn't make sense to use the Henderson-Hasselbalch equation, because then you'd have to plug in a 0 into the Henderson-Hasselbalch equation, and the Henderson-Hasselbalch equation won't give you reasonable answers when you plug in the 0. Okay. If we started with both the weak acid and the conjugate base, that's when we use the Henderson-Hasselbalch equation. That's when we don't need to use the start, change, end table. Now, let's see. Where did you get this? If you can see what I highlighted here. Where did this come from?
Oh. Oh, wow. I was referencing another problem just to see how I did it. And I think I accidentally wrote that number instead of 5 times 10 to the negative sixth.
So what should be written here?
5 times 10 to the negative sixth.
And now, why? Where would that come from? What's the logic behind that?
Because we don't know how much concentration we're losing in the reaction, so we denote that with X. So we have the concentration minus how much is being lost.
Okay, so tell me again, what would you write here then?
5 times 10 to the negative 6 minus X.
Okay, that's right. In that case, why did you write here just 5 times 10 to the negative 6?
Because we have it's the products over the reactant. which equals the Ka.
That's right, but why wouldn't this then be 5 times 10 to the negative 6 minus x, since we just said that we ended up with 5 times 10 to the negative 6 minus x of the reactant?
Because the x is so insignificant.
That it's approximately the same as the 5 times 10 to the negative 6. Okay,
Yep.
That's right. I just want to make sure we're understanding the steps so that we can adapt rather than just doing these mechanically. Okay, so then this came out to be correct here. But again, it's good to make the table make sense so that you can adapt to other types of problems. All right. I noticed that you rounded this off to only 1 digit. And that as a result, your answer was somewhat inaccurate. When you're taking the test, you should round off to maybe 2 or 3 digits. At least 2 digits might be good enough, but 1 digit is usually rounding off too much. Okay. In any case, that was the right answer for that. Let's look at number 2. Okay, so this was the correct method for this problem, and you correctly wrote that it was a strong acid problem. Good. But what type of problem was question 1?
Question 1 was a weak acid problem.
Good. So I just wanted to point out what you wrote here turned out not really to be the best way to put this. You said it was weak acid plus conjugate base. But it's better to say it's just a weak acid problem because we only started with the weak acid. I think you probably said and conjugate base because we ended up with both the weak acid and the conjugate base. But it turns out that when you're classifying the problems, it's more useful to classify them based on what you started with, because that determines how you solve the problem. How you solve the problem is based on what you started with, not on what you ended up with. Okay, so both of those were correct. pH is the acidity of the acid, pKa is the acidity of pH is the acidity of the solution, pKa is the acidity of the acid. Okay, that's a good answer. Good, that's good that you were remembering that. It's number 4. Did you answer number 4 from memory?
Yes.
Good, because that's what I think you'll need that on the test. Oh, based on my memory, those look correct. Number 5.
I think I gave you another picture of the bottom part is 6.
Okay. Well, this picture came out much more clearly. All right. Or maybe not that clear. Let's see, what did you write here? Electrostatic interaction is the attraction between positively charged atom and a negatively charged atom. Okay, and in your picture, did you show an electrostatic interaction between main chain or side chain atoms?
Side chain atoms.
Okay, good. So that was 6A. It looks like you sent some of these multiple times. Hydrogen bond is a weak attraction between a delta positive hydrogen and a delta negative atom. can you describe to me what's the order of the atoms on the main chain? What atoms make up the main chain of an amino acid?
The terminal N, the alpha carbon, the carbonyl carbon, and then the phi bond, then the nitrogen, then the psi bond, and then again with the alpha carbon.
Okay, that wasn't 100% correct. That was on the right track. Let's just describe one amino acid. So there's 3 key atoms in one amino acid's main chain. What would be the 3 key atoms in one amino acid main chain?
Carboxycarbon.
What, and list them in the order that we would usually list them.
The amide.
Or
the alpha carbon.
I guess what I'm going for. Do you remember the slogan that I've mentioned for memorizing those 3 atoms? I don't know if you know what I'm talking about. What's the slogan for the 3 atoms?
I feel like I know what you're talking about. But
what I'm going for is, I think it's helpful to memorize a slogan. Nitrogen, alpha carbon, carboxycarbon. Because if you don't memorize this slogan, people tend to make mistakes. Nitrogen, alpha carbon, carboxycarbon. What comes after the carboxycarbon?
Nitrogen.
Another nitrogen in the next amino acid. So then nitrogen, alpha carbon. This is not the whole picture, of course. This is just a really helpful slogan to make sure you're getting the chain correct. Nitrogen, alpha carbon, carboxycarbon. So let's look at this. Did you get the correct atoms for this amino acid? Nitrogen, alpha carbon, carboxycarbon.
Yes, not the bottom one.
Yeah. So what's wrong here at the bottom?
It should be nitrogen, alpha carbon, carboxycarbon.
So for one thing, this is in the wrong place.
Yes.
This is not in the right place. And we shouldn't just have a little oxygen all by itself over here. So this is not a real amino acid. So this is an important skill. I think on Monday's test, you're going to have to draw some peptides. On Mondays, that's why I'm assigning you these questions. I think you'll have to draw some peptides. So you want to be able to make sure that you're getting that correct without those mistakes. It's easy to make those. So what I find, I tend to make mistakes unless I memorize that slogan. So to review, what's the slogan we've been talking about for the main chain atoms?
Nitrogen, alpha carbon, carboxycarbon.
Whenever I write a main chain, I always say that under my breath, because otherwise I'm going to make mistakes. And you can see that's kind of what happened here. All right, so this main chain isn't quite right. To save time, I'm not going to rewrite it with you right now, but it's important to see how to write that correctly, because I think you could definitely have to do that on the test. All right. What you wrote up here was correct. What would go after this carbon?
Nitrogen.
That's right. But it's okay that you left it out because you were just focusing on one amino acid. So it's perfectly okay to leave out that nitrogen here. In fact, it's probably better since you just wanted to draw that one amino acid in the chain. So let's check what you wrote here. Are these atoms correct?
Yes.
Now, did you draw one complete amino acid here, or 2 complete amino acids, or kind of like 1.5?
1.5.
Which I don't, so I think it would be better to draw a complete number of amino acids. You might not have thought about that here, but it would have been better to draw maybe 2 complete amino acids here if that's what you wanted to do. Since you wanted to use this nitrogen, it would be better to draw the rest of the amino acid that's connected to it. Are these atoms correct?
No.
No, they're messed up. Okay. All right, a van der Waals force. This is a little hard to read. Van der Waals force are when the electron. Why don't do you have it in front of you? Why don't you tell me what you wrote down?
Yeah. The van der Waals forces are when the electron positions are random between a hydrogen and a carbon. The hydrogen temporarily gains a delta negative charge or a delta positive charge. And then I put a second definition, a simple one. A weak interaction between a temporary delta positive or temporary delta negative atom.
Okay, that second definition is probably the most practical one. Yeah, weak interaction between a temporary delta positive and a temporary delta negative. That's right. Your first definition is on the right track. The electrons are random. So the electrons are always random. So that first definition is not quite right. Electrons always fluctuate at random, but the van der Waals force comes when the when the electron density happens to randomly bunch up in one place. It's a little bit hard to put into words. So van der Waals forces occur when the electrons randomly form temporary delta positives and delta negatives. Might be a good way to put it. The van der Waals force occurs when the randomly moving electron density forms temporary delta positives and delta negatives. It's usually between hydrogens and carbons, although it doesn't necessarily have to be. But that's probably what you'll usually see in this course. What amino acid did you use here for this van der Waals force?
Glycine.
And down here?
I can't see that actually. I used glycine also.
Oh? You're saying this is glycine too?
It's not correctly drawn, but I try to do the two hydrogens.
I see. Okay, that certainly would be positive. It looks more like you drew it, like the way you drew it, I think it looked more like alanine. All right, now one problem with that is, are van der Waals forces strong or weak?
Weak.
Weak. So they're usually only going to be significant if there's quite a few carbons or hydrogens to interact. So it would have been better to have drawn side chains that are bigger, so there's more carbon-hydrogen interactions. I don't think I really quite explained that last time. What would be some side chains that have, what would be some non-polar side chains that are bigger?
Proline.
That would be an excellent one to use then for van der Waals interactions. What's another one that's bigger?
Isoleucine.
Yeah, so those would have been better because then you can have van der Waals interactions at more than one point. If you only had a van der Waals interaction between two atoms, it would be so small as to be insignificant. Van der Waals interactions are only significant if you kind of have at least a bunch of carbons and hydrogens opposite each other, so there can be a bunch of points of overlap. The way I described it was a little unrealistic. Usually it's only significant if you can get it between a bunch of side chains, but that would be a little bit tedious to draw. This was on the right track. So why don't we do one of these over again? Why don't we do the hydrogen bond one on your screen? Try to, again, draw a peptide, draw hydrogen bonding between different parts of a peptide on your screen. Let's try to be a little bit more careful to draw the main chain atoms accurately.
With different molecules, right? With different amino acids.
I don't care. Just as you can use the same one if you want, just as long as you're drawing the main chain more accurately. That's the main thing I wanted to look at. We'll do it for hydrogen bonds. Like I said, it's hard to draw the van der Waals forces because the van der Waals forces are usually only significant when there's van der Waals forces in a bunch of different locations, a bunch of different neighboring locations. So let's try the hydrogen bond.
Okay. So, 1 second.
So what we're trying to do here is we're trying to redo problem 6, part B. Problem 6, part B.
I put an NH3 here. Okay.
Okay, now remember, one of the main things we were trying to do, one of the main reasons we're redoing this is because we're trying to draw the main chain atoms more accurately. What's the slogan for making sure you get the correct atoms on the main chain?
Nitrogen, alpha carbon, carboxycarbon.
All right, so do you have that correct for the top amino acid?
Yes.
Do you have that correct for the bottom amino acid?
The fact that it's upside down is confusing.
Okay, well, you shouldn't so much think of it as upside down. Well, the point is, remember, it's just like a bunch of railroad cars that are all connected to each other. So the order should be the same. It's not so much upside down as that it's maybe turned into reverse. But if we're visualizing, I can see how it's a little confusing at first, but if we visualize it clearly, we should be able to figure out what the order is.
Okay, so there should be a nitrogen?
Where?
Here?
I guess we're not really visualizing this correctly. Maybe one moment. All right, can you see my screen?
Yes.
So what is the slogan?
Nitrogen, alpha carbon, carboxycarbon.
Nitrogen, alpha carbon, carboxycarbon. Nitrogen, alpha carbon, carboxycarbon. Nitrogen, alpha carbon, carboxycarbon. Nitrogen, alpha carbon, carboxycarbon.
Okay.
Nitrogen, alpha carbon, carboxycarbon. Nitrogen, alpha carbon, carboxycarbon. Okay. So do you see what I meant when I said it looked like you weren't quite visualizing what you were doing here? Up here, the nitrogen is on the left and the carboxycarbon is on the right for each amino acid. But if we make a U-turn over here, if we're going to keep the same order, now the nitrogens are on the right and the carboxycarbons are on the left. That's the only way to maintain that same slogan. Nitrogen, alpha carbon, carboxycarbon. Nitrogen, alpha carbon, carboxycarbon. Nitrogen, alpha carbon, carboxycarbon. Now the nitrogen here are on the right because we made a U-turn. Nitrogen, alpha carbon, carboxycarbon. Nitrogen, alpha carbon, carboxycarbon. Now, for the problems, oftentimes if you're trying to draw a whole strand, it's convenient not to draw every single amino acid like this. You don't always want to draw every single one. I just drew all the amino acids here so you can see what the pattern is. Is that making any sense?
Yes.
Okay, let's go back to your screen then.
So do you want me to draw on the nitrogen?
Well, I just like us to keep working on 6B so that we draw an accurate picture.
Okay.
All right. Now remember what type of interaction, so let's see, do we have the main chain here correct? Yes. Now you didn't have to draw these two, you drew the interaction here between the N terminus and the C terminus. You didn't have to do it that way, but that's okay. Do you remember what type of interaction were we trying to draw here?
Van der Waals?
Oh, okay. Actually, I'd asked for a hydrogen bond, but okay, so here we have a van der Waals interaction. Now, if you were really going to do the van der Waals interaction, I guess I'll go back to my screen and show that in a moment. So let's do another one with the hydrogen bond. The hydrogen bond's a little bit easier to show.
Do you want me to do it with any specific, like the side chains, or
whichever
does
is
it
the
matter?
easiest for you. Yeah, whichever you like.
No, no. Okay. Yeah, well.
Okay, so that's good that you took your time and got the right order of the atoms on the main chain. What's the slogan for the atoms on the main chain again?
Nitrogen, carboxycarbon, alpha carbon.
Say that again, please.
Nitrogen, alpha carbon, carboxycarbon.
That's right. That's actually not what you said a moment ago. So it's important to get those exactly right. So to review, let's say it one more time.
Nitrogen, alpha carbon, carboxycarbon.
Okay, that's very important. Like I say, I think it's likely you'll have to draw something based on that on the test. Now, what type of interaction were we trying to draw here?
This is a hydrogen bond interaction.
What's the definition of a hydrogen bond?
It's a weak interaction between a delta positive hydrogen and any delta negative atom.
Now, what's the delta charge on your oxygen there? Delta positive, delta negative, or neutral?
Delta negative.
You can go ahead and draw that in. How do you know it's delta negative?
Because it's a highly electronegative atom.
Let's see. That's on the right track, but highly electronegative elements don't always have delta negatives. The key is that you have to compare it. Why would a highly electronegative element have a delta negative charge?
Because it's pulling the electrons in the double bond closer to it than the carbon.
That's right. So the point is not that it's so much that it's highly... so the point is that it's more electronegative than the carbon. Okay, so how about on the hydrogen? What's the delta charge on the hydrogen? Delta positive, delta negative, or neutral?
On the hydrogen, it's delta positive because it's not pulling... It's neutral because it's with a carbon.
That's correct. So who's more electronegative, the carbon or the hydrogen?
They're similar.
That's right. So are the delta charge, is the hydrogen delta positive, delta negative, or neutral?
Neutral.
So can that hydrogen form hydrogen bonds?
No.
Okay, so it's good that we're reviewing this. This is very important. I think I might not have quite been clear about this last time. Can all hydrogens form hydrogen bonds?
No.
No. What does a hydrogen need to form a hydrogen bond?
Hydrogen needs to be delta positive.
That's right. Only delta positive hydrogens can form hydrogen bonds. That's actually quite important. What's the name of the amino acid that you drew up on the top?
That is valine.
That's right. So can the valine side chain form hydrogen bonds?
No.
No. Why not?
Because the hydrogens aren't delta positive.
That's right. Because it doesn't have any delta positives or delta negatives. Okay, so it looks like we didn't get that right. So let's try again.
Okay.
Okay. So did you end up drawing the hydrogen bond? So what type of interaction did you draw?
Hydrogen bond.
That's right. Between main chain or side chain elements.
Main chain elements.
Okay, that's fine. So this is correct because now the hydrogen does have a delta positive. So this is a very important skill to be able to understand these. And you drew complete amino acids, 2 complete amino acids on the top and 1 on the bottom. So that's better. I won't spend the tutoring time on this, but it would be a good exercise to make sure you can draw hydrogen bonds between side chains. There are some side chains can form hydrogen bonds and some can't. So that would be a good exercise. But for now, we'll just see those main chain interactions. Okay, so it's very important to keep in mind, can all hydrogens form hydrogen bonds?
No.
Now, what does the hydrogen need to form a hydrogen bond?
It needs to have a delta positive charge.
That's right. So that's very important. Can you see my screen? Hello, can you see my screen?
Yes. Yes.
Okay. I just want to make one little minor point. I noticed that when you were drawing the valine, you were doing it like this. I just wanted to point out, TAs might not like this. When you're drawing bond line notation, you don't need to show hydrogens, or you can just show one hydrogen. But if you actually draw in the letter for the C, you have to show all the hydrogens. That's just the convention. So you can either use bond line notation, and then you can show as many hydrogens as you like, or none of them. But if you write in the letter C, then you actually have to show all the hydrogens. So it would be incorrect to do this. You can't write the letter C and only write one. Maybe the easiest thing you could have done was this then. Now you're still using bond line notation, so it's okay to show only one. The other thing I wanted to point out is, again, van der Waals forces are usually only significant if there's multiple points of overlap. So if you were going to show van der Waals forces, it would be probably better, if you can see what I'm doing, to do it like this. Van der Waals forces are usually only significant when there's a bunch of points of overlap between the compounds. So it's best for the van der Waals forces to show it like this, to show that there's a bunch of points of overlap, rather than just 1 point overlap. If there's only 1 point of overlap, then the van der Waals force would be too small to be significant. Usually the van der Waals force would involve multiple side chains, too, not just one. Okay. Okay, I'm not seeing an answer for number 7. Do you have that written down? You can just tell me what you wrote down for number 7.
What was number 7? Oh, I'm sorry. What is that? Oh, I think I might have skipped that one.
All right, well, no time like the present. Let's answer it now. You can just tell me aloud, what is the hydrophobic effect?
The hydrophobic effect is, I don't remember off the top of my head. Hold on a second. I don't remember.
Okay, now we should review that. So that material we went over last time, all those types of interactions are going to be very important for the course.
Oh, now I remember. I needed a few more seconds to think of that. Is it when you have like a non-polar molecule and like polar solution, and the non-polar atoms are together, and you got the surface area of water to there's less water bonding, or there's less water surface of the non-polar object.
Okay, well, that's actually, from going from not remembering, that's actually an excellent definition. Yes, so that's actually a very good subtle definition. Very good. So the idea is, what happens to non-polar substances in a polar solvent? In water. They clump together. They clump together. We can just focus on water because that's what you'll be focusing on. That's right. So the hydrophobic effect is that hydrophobic substances clump together in water to reduce their surface area. And you explained why that is. The water would rather border on water instead of bordering on the hydrophobic substances. So the water is going to push the hydrophobic substances together so that they have less of a border with the water. Okay, so that turns out to be quite important. Now the hydrophobic effect is a little bit subtle. Remember, so the nonpolar substances clump together. Is that because the nonpolar substances are attracting each other? Well, nonpolar substances do have van der Waals forces, but the van der Waals forces are pretty weak. So remember, the van der Waals forces are not the main reason they clump together. The main reason they clump together is to reduce their surface area with the water. So it's not so much that the nonpolar substances are attracting each other. Their attractions are pretty weak, but they're being pushed towards each other by the water, basically. The water pushes the nonpolar substances towards each other so that the water will have less border with the nonpolar substances. It's a little bit of a subtle idea. But the basic idea is that the nonpolar substances clump together. Okay, so that's important. I think maybe you skipped number 8 as well. I didn't see any answers for that. Let's try answering number 8 now, because that's an important question. What do you think is the answer to 8A?
What force or I don't know why I skipped this. What force or interaction maintains the shape of the alpha helix? It's the hydrogen bonds and hydrogen bonds.
Excellent. Hydrogen bonds between the side chain atoms or main chain atoms?
Between side chain atoms.
Actually,
main chain, sorry.
It turns out to be main chain. I don't know if I emphasized that enough last time, but secondary structure let's make sure we have this in your notes. Secondary structure is maintained by hydrogen bonds between main chain atoms. I don't know if you remember, but actually last time I actually drew you a picture. I showed how each carbonyl oxygen will be hydrogen bonded to the nitrogen 4 residues further down the chain. I don't know if your instructor expects you to have it memorized in so much detail, but in the previous session I drew a picture of how each carbonyl oxygen is hydrogen bonded to the nitrogen 4 residues down the chain. You might not be tested in that much detail, but we should know that the secondary structure is maintained by hydrogen bonds between main chain atoms. What are the types of secondary structure?
Beta-pleated sheet and alpha helix.
Yeah, those are the main types that we're focusing on. So we've only, both of those are maintained by hydrogen bonds between main chain atoms. We haven't really talked much about the beta-pleated sheets yet. Okay, that's important. What force or interaction, so what is secondary structure maintained by?
It's maintained by hydrogen bonds.
Yeah, between main chain atoms. What about primary structure? What's primary structure maintained by?
Primary structure is maintained by covalent bonds.
That's right. And what do we call the bonds between amino acids in the primary structure? What do we call a bond between 2 amino acids?
A bond between 2 amino acids, it's a polypeptide bond. Peptide bond.
Peptide bond. Peptide bond. And which of the 4 types of interactions is a peptide bond? Covalent, van der Waals, hydrogen bonding, electrostatic?
Covalent.
That's right. Okay, those are all important. All right, how about 8b?
8b says, what is the value of the rise of an alpha helix? It's 1.5, is it 1.5 excuse me, 1.5 angstroms per residue.
Excellent. Those are good units for solving problems. 1.5 angstroms per residue. How about C?
C, how many residues in one turn? 3.6.
What would be the units for that?
3.6 residues.
Let's see, good. Although I think maybe better residues for solving, better units for solving problems would be 3.6 residues per turn. Because if there were 2 turns, there would be more than 3.6. How about C?
How long is one turn of an alpha helix? One turn of an alpha helix is um, 3.6 times 1.5. And that is 5.4 angstroms.
What would be the units for that?
Angstroms.
Good. Although better units would be 5.4 angstroms per what?
Per residue.
No.
5.4 angstroms per turn.
That's right. Do you see how that's different than per residue?
Yes.
All right, so it's 5.4 angstroms per turn. Okay, but that was a good method for solving C. So you don't need to have C memorized. If you have A and B memorized, you can figure C out, just like you just did. So that's very good. Okay. Angstroms are a measurement of length. Here's the symbol. Excellent. I think we should know how long an angstrom is, though. Do you remember what the exact length of an angstrom is?
1 angstrom is... I don't...
Okay. I think I didn't emphasize that last time. Take a guess.
Is it 0.01 nanometers?
Close. It's 0.1 nanometers. That means 1/10 of a nanometer. I think the best thing to memorize is 10 to the negative 10 meters, or maybe all of these would be good. It's 1/10 of a nanometer, 10 to the negative 10 meters. This is the unit of length that's going to be used in your biochem course, so I think we should memorize what it means. So this is what an angstrom is. All right. Number 11. No, number 10. Okay, you got the correct answers here for number 10. There's a couple of different ways to solve this, but it looks like your method worked. You got the correct answer for number 10. Good. I'll look at number 11. All right, this is the right answer. Now the turns here don't. It looks like you approximate you rounded your answer for the turns off to an integer. But what I actually got when I solved this was about 8.8 turns, and we don't need to round that off. You can have a fraction of a turn. It just means that it turned around completely 8 times, and then it started turning around one more time, but only got 8/10 of the way. So probably it would be good to round this off a little bit less and leave an extra decimal point. That answer still makes sense. But anyway, it looks like you used the right method here for number 11. So let's look at number 12. .6 angstroms and all right, so it looks like you were right on track for that. That's correct. Okay, so questions 10 through 12, unfortunately I don't have any practice tests from your instructor, so it's a little hard to guess what they're going to put on the test, but questions 10 through 12 are pretty typical of biochem problems. I don't know whether how much your instructor expects you to have memorized. You could ask, but you might need to have those numbers memorized, the 3.6 and the 1.5, say, in order to get this type of problem right on the exam. Okay, so it looks like you did that correct. All right, so let's look at number 13. Proteins can be enzymes, receptors. They can have structural roles. The rest of it I can't really read here. What else did you write?
Hormones, antibodies, transport, and motor.
Okay. Was that from memory?
Yeah.
Okay, good. There's probably a bunch of other things that we didn't talk about, but proteins have lots of structures. The most important is enzymes, but there's many different functions, many of which you'll study in the course. Oh, here's a clearer picture. Let's see. Did you get to number 14 and 15? I think maybe you didn't send those to me.
I didn't get to them. I wasn't able to.
Okay, well, let's talk about that now. How would you answer number 14?
Number 14. So what type of molecule is alpha-keratin? It's a protein molecule.
That's what I was going for. Yes. How about B?
The function of alpha-keratin, structure.
Good. But structure of what? What types of
nails, skin, teeth, bone, or horns, claws, hair.
Like this?
Hair. Yes.
All right. Let's skip down to collagen. What's the function of collagen?
That's for collagen. That is collagen. Sorry. Keratin. I forgot what keratin is.
Okay. Actually, this is kind of mixing up the keratin and the collagen. So let's try to split these apart. So when I'm trying to memorize keratin, the first thing I think of is hair. And then collagen, maybe I would start with bones. So what are some other things that are somewhat similar to hair that we might put in this list?
Wool.
Yeah, that's an easy one.
Nails, claws, and horns.
Obviously, nails and claws are similar. And it turns out that horns are made of kind of hair or hair-like material. Okay, good. I think maybe horns really are just hair. Okay. So then what would go down here for collagen?
Skin, cartilage, tendons, and teeth.
Okay. I don't know. So hopefully you can see some similarity between these. Bones and cartilage play somewhat similar roles. Teeth, somewhat similar to bones. Tendons. Okay. So I think there's a good chance that you'll need to know something about these two lists for the exam. What roles these are both play structural roles, but in two different types of tissues. Okay. So very good. Let's go back to 14 and try part C for number 14.
The structure of alpha-keratin is a double helix. It's a it's a double helix.
Pretend that was a test question. So what would you say for 14C if it was a test question?
It contains two helix coiled together.
Okay, that will give you some credit, but you didn't say what type of helix.
Alpha helix.
All right, that's important. So for full credit, so let's try again. What would be a better answer on the test for 14C?
Two alpha helix coiled together.
That's right. That's the basic structure. And then, so that's called a coiled coil. And then remember, oftentimes those coiled coils will be further coiled around other strands of alpha-keratin to build up strength. So the basic structure is a coiled coil of two alpha helixes coiled around each other. Here's the basic structure. But remember that this structure itself could be further coiled around other coiled coils to build up strength. Just like you can you can twist strands of rope around each other or strands of thread around each other to build up their strength. You can take one of these coiled coils and take another coiled coil and coil them around each other to get a higher level of strength. So here's the picture your instructor gave you. Here's one alpha helix. Here's the coiled coil of two alpha helixes. But notice here how they're taking, here they have a pair of coiled coils, and now you can see they have a whole bunch of coiled coils all twisted around each other. And you can see how that would kind of increase strength, just like you could increase strength of a rope by having a bunch of I don't know, threads or strings all coiled around each other. Okay. But the basic structure is this, a coiled coil of two alpha helixes. All right, let's see where we were here then. What's holding these two coiled coils together? What's holding
hydrogen bonds?
Say again?
The hydrogen bonds in glycine.
Hydrogen bonds in glycine? I think you might be confusing a couple different ideas together. Actually, that was kind of a rhetorical question because I don't think we talked about that last time. Oh. Okay, so what's holding these together could be van der Waals interactions or ionic interactions between the two coils. So I don't think we actually talked about that last time, but the two helixes could be linked by van der Waals interactions or ionic interactions between the coil. Maybe hydrogen bonds too. Just there's various weak interactions. We talked about the weak interactions between these. Now, something else that's very important, though, that might be holding those together, these two coils could be linked by disulfide bonds. Now, I don't think you and I have talked about disulfide bonds yet, but they're certainly very important, so now would be a good time. Have you learned about disulfide bonds in class?
No.
Oh, okay. Well, I'm sure your instructor's talked about it.
Oh, no, but she has.
But maybe you don't remember what they are?
Yeah.
Okay, well, let's talk about that now. All right. So do you remember anything about them? What do you know what types of amino acids form disulfide bonds?
It's ones with sulfur groups, like the sulfur, well, it would be cysteine and methionine, maybe.
That's a good start. So let's talk about that some more.
Cysteine, I know, does.
What's the name of this side chain? Or this amino acid?
Cysteine.
They're both cysteine. Yes. Okay. I shouldn't have drawn this so big. Okay. So, yeah, these can form the side chains. The disulfide bridge. I shouldn't have drawn that so big, but... Okay, I think I got it right now. So do you see the difference between here? Here's a regular, this is, what's the name of this amino acid again?
Cysteine.
Cysteine, good. And here we have a disulfide linkage between two cysteines. So disulfide linkages occur between two cysteines. Okay, you can tell me when you have that down.
Okay.
So we've gone over 4 different types of forces or interactions. Which of the 4 different types of forces or interactions is a disulfide linkage?
Strong.
Yeah, I haven't quite spelled this out, but the 4 types of interactions we've talked about are covalent bonds, electrostatic interactions, hydrogen bonds four types of interaction. Which of these are strong and which are weak?
Covalent bonds are strong and the rest are weak.
Okay. Tell me when you have that down.
I have that down.
So which of the four types of interaction is a disulfide linkage?
Strong.
Yeah, but which of the four types?
Oh. Covalent.
Yeah. You can see that because I drew it as a solid line. And the only reason it's a long solid line is because these are far apart on my page. It's just a regular covalent bond. Disulfide linkage is just a regular covalent bond here. Okay, do you remember the ideas of being oxidized and reduced that we talked about in organic chemistry? Do you remember if something is very oxidized, what does that mean? Or if something becomes more, what's happening to something if it's being oxidized?
It's losing something. Oxidized oil rig is what I remember, so it's loss of electrons.
Okay, that's right. That definition is usually more, that's a good definition. That definition is usually more useful for general chemistry than for organic chemistry or biochemistry. So something is oxidized if it loses bonds to electronegative elements. And gains bonds to hydrogen. Something is reduced if it gains bonds. I got that backwards. Let's try again. Oxidized means you gain bonds to electronegative elements, lose bonds to hydrogen. Reduced means you lose bonds. To electronegative elements. Gain bonds to hydrogen. This definition is usually going to be more useful to us than the electron definition, I think, in this course. You tell me when you have that down.
Okay.
So, is this picture the oxidized or the reduced picture?
That is the reduced picture.
Because they have the extra bond to hydrogen. Looks like I still didn't draw that correctly. All right, so here's the correct picture of cysteine here. And is this the oxidized or the reduced picture?
Oxidized.
Because they lost the bonds to hydrogen, and each sulfur gained a new bond to another electronegative element, another sulfur. So how do you form a disulfide linkage? Do you form a disulfide linkage by oxidizing cysteine or reducing it?
Say that again.
How would you form the disulfide linkage? By oxidizing cysteine or reducing it?
Oxidizing.
Yes. And how would you break a disulfide linkage?
By reducing it.
That's right. Okay, so what type of thing would break a disulfide linkage? An oxidizing agent or a reducing agent?
To reducing?
Because we want to, the way to break it is by reducing it. We'll see that soon. Okay, so here's a disulfide linkage. Did I draw this disulfide linkage? So the disulfide linkage is always between the side chains. Is this disulfide linkage between two parts of one peptide or between two different peptide strands?
One peptide strand.
But you could also form a disulfide linkage between two separate peptide strands. So is this between two? What type of interaction do we? What type of bonding do we have here?
Also disulfide bond.
Yep. Although I screwed up and didn't draw it correct again. Let's try again. It's a disulfide bond. That's right. Which of the four types? Okay, that's good. Disulfide bond. Is this between two strands or one strand?
Two strands.
It kind of looks like two strands, the way I've drawn it here. Okay, so disulfide linkages could be between two parts of one strand or between two separate strands. Are disulfide linkages strong or weak?
They're strong.
Because they're covalent. Okay. Okay, so that's a very important topic that we haven't talked about before.
Do they only occur with cysteine?
That's a good question. So the other one that, what's the name of this side chain?
That is methionine.
It also has a sulfur. Can you think of any reason why this would have a hard time forming a disulfide bond?
Because it's surrounded by two carbons.
Instead of a hydrogen. Remember that things are usually reduced by gaining bonds. Things are usually oxidized when they lose bonds to hydrogen. Now, that's not always true. There are oxidations where you break a bond to carbon, but those are usually harder, and that's not what's going to happen here. So no, methionine does not form disulfide bonds. That's very important. Because it has a sulfur with a methyl rather than a sulfur with a hydrogen. Cysteine is the only amino acid that forms disulfide bonds. Cysteine is the only amino acid that forms disulfide bonds. Okay, do you have that in your notes?
Yes.
All right. All right, that was a little bit of a digression. And now I wanted to go back to describe the structure of alpha-keratin.
Alpha-keratin has two alpha helix coiled together.
Yeah, it's a coiled coil of two alpha helixes. That's the basic structure. Now, what's holding those two alpha helixes to each other? Well, there could be weak interactions holding them together. But also, there could be disulfide bonds between the two coils. So that's a good thing to have in your notes. You should put that in your notes. Where were we? So here's our structure for alpha-keratin. Should be the word other. You can tell me when you have that down.
Okay.
What are the weak interactions?
Hydrogen bonds, van der Waals forces, and electromagnetic interaction.
Yeah, there could be any of those holding the two coils together. Also, sometimes there are disulfide linkages between the two coils. All right. Define a disulfide link.
Disulfide link is when a cysteine bonds to, or it's an oxidation of a cysteine with another cysteine.
Two cysteines are oxidized to form a covalent link between them. Okay, good. Are disulfide bonds strong or weak?
Strong.
So let's talk about something that your instructor talked about then in the notes that you sent me. Let's say you are analyzing an alpha-keratin with many disulfide bonds. Would you predict it then to be relatively rigid or flexible?
Rigid.
Because those are strong bonds that hold the structure in one position. Weak interactions can easily form and then reform. Weak interactions can be easily broken and then reformed in a different pattern. But disulfide bonds are more difficult to break, so they're more likely to be rigid. So what type of substance is this more likely to be? Can you remember what are some of the biological structures that have alpha-keratin?
Some biological structures are hair, wool.
Anything else?
Horns.
Good. I think horns are just made out of hair, but that's good. Anything else?
Teeth.
Now, teeth is in the collagen list.
Collagen.
Do you see how teeth are kind of more like bones?
Yeah.
So the big one you're missing is nails and claws. Turns out that nails and claws are actually more similar to hair in structure than they are to teeth and bones. Now here's my question. Which type of alpha-keratin is this particular alpha-keratin here, is this more likely to be found in hair or nails? If the alpha-keratin has many disulfide bonds, is it more likely to be in hair or nails?
In nails.
Because
nails are more rigid than hair.
Okay, it's just that simple. Yes. So the point is, so that's something you should have been wondering. When I told you that alpha-keratin makes up hair and nails, you should have said, that sounds bogus. Hair and nails are totally different. How can they be made of the same thing, right? I mean, hair seems very different from nails. Hair is quite flexible and windy. Nails are quite rigid. So you should have been wondering, how can they both be made out of the same type of molecule? Well, here's the answer. It's the same type of molecule, but one version has more disulfide bonds. Of course, I'm sure there's other differences, but one key difference is nails are made out of alpha-keratin with more disulfide bonds, which makes them quite rigid, whereas hair is more flexible because it has fewer disulfide bonds. All right, I think that would make a good type of test question. Especially because it's right here in your lecture notes. If you did a chemical analysis, who would have a higher sulfur content? Fingernails or hair?
Fingernails.
Because
it has more disulfide bonds.
How do you know?
Because they're more rigid.
Okay, so I'm looking here at your lecture notes that you sent me. So one of the things that you wrote down that your instructor said was fingernails have a higher sulfur content than hair. When you wrote that down, you might not have been quite clear why your instructor was emphasizing that, but now hopefully we see the reason why your instructor was emphasizing that fingernails have a higher sulfur content than hair is because it's related to the issue of the disulfide bonds. So that would make a good test question. Okay. All right, so I think that's some of the key things that your instructor stressed about alpha helixes that might be on the test. So now let's try question 15 from the homework. What would be the answer to 15A?
Okay. What type of molecule is collagen? It has three. It's not. I know it's not alpha helix. It's three peptide strands coiled together.
What would be a good name for a structure with 3 strands coiled together?
Triple helix.
Excellent. Good. So it's 3 peptide strands coiled in a triple helix. None of the strands are alpha helixes. Why does your instructor keep emphasizing that none of them are alpha helixes? Well, just because some students are lazy and they just think wherever they see the word helix, they think it has to be an alpha helix. Well, alpha helixes are just one type of helix. So none of that. So this is a little bit of a digression again. Now we're digressing from alpha helixes and talking about collagen. So that's 3 peptide strands coiled in a triple helix. Okay, good.
None of the strands.
Okay. So they're tied together. The 3 peptide strands are held together by hydrogen bonds.
So for keratin, though, the shape is alpha-keratin, right?
One moment. The only type of keratin that your class has talked about is alpha-keratin. Yes. The only type. So I might have been a little sloppy here. I guess I should have said alpha-keratin. All the examples we talked about were alpha-keratin. I think there's another type that's called beta-keratin, but your course is not studying that. So alpha-keratin is the compound that makes up hair, nails, claws, and it's the one that's made up of coiled coil of 2 alpha helixes. I don't know, maybe it's called alpha-keratin because of the alpha helixes. I'm not sure. I think there's another type of compound called beta-keratin, but your class hasn't talked about that. I think beta-keratin might have some beta-pleated sheets in it. I'm actually not too familiar with that. That's actually not usually covered too much in the course. So anyway, yeah, all the keratins we've talked about, the correct name for them is alpha-keratin. Did that answer your question?
Yes.
Okay. Let's see. But if your instructor just uses the word keratin on the test, I'm pretty sure they mean alpha-keratin, because that's the only type that I think your instructor is covering. All right, so back to collagen. Let's see. Okay, so we also talked last time about some of the components of collagen. What can you tell me about the primary structure of collagen? What can you tell me about the primary structure of one of the peptide strands?
It doesn't have a pattern of every 3 glycine.
That's what I was going for. Good. That's a pretty firm pattern. I don't think there's really any exceptions to that. So each strand has this pattern. For collagen. Do you remember why each third amino acid has to be glycine? Where does the glycine go in the overall structure? Where is it in the triple helix?
In the interior.
Why is glycine the only thing that we could put in the interior?
Because it hydrogen bonds with other glycine.
That's actually a mistake. That's not correct. First of all, what's the name of this amino acid?
Oh, I know. That's glycine.
Can glycine form hydrogen bonds?
No, it cannot.
How can you see that it cannot?
Because it has no delta positive hydrogens.
That's right. So I think there was a misconception there. Why did you say it could form hydrogen bonds? I think you probably got a couple things mixed up. You remembered that glycine side chain is a hydrogen. The glycine side chain is just a hydrogen. So you might think, gee, if it's just a hydrogen, it should be great at forming hydrogen bonds. But it doesn't work that way because hydrogen bonds, so hydrogen bonds is really not a good name. They would be better to call them hydrogen delta positive or delta positive hydrogen bonds. So it would be better to call hydrogen bonds delta positive hydrogen bonds. So regular hydrogens, like in glycine, can't form hydrogen bonds. So I think there was a misconception there. All right. Any other ideas why glycine has to go, where glycine, I forget what your first answer is. Where is the glycine in the triple helix?
In the interior.
Yeah, why? What's special about glycine side chain? It can't form hydrogen bonds. What's interesting about it?
It's tiny. It's small and flexible.
Yeah. Which of those is important here?
Small.
Yeah. So it's the only one that there's room for in the interior. Because the interior is where all three helixes are butting into each other. When all three helixes are butting into each other, the only thing there's room for is a small glycine. Here's the picture we looked at last time. So remember, the glycines would be in the center here. Notice that in the center, there's just not that much room. If you tried to put a proline in the center, that would separate the two strands so they couldn't lie flat with each other. Or in fact, if you try to put any side chain in the center besides glycine, that would separate the two strands, the three strands, so they couldn't be close to each other anymore. So the triple helix has to be packed quite close. The point is the triple helix is packed quite close, so the only thing there's room for in the center is the glycine. So you can see it looks like there's a turn, maybe there's a turn every three peptides here. So every third one goes back to the interior. Okay, do you remember anything else about the primary structure of each strand? Anything besides the fact that every third amino acid is glycine? Remember anything about what often goes in the other two positions?
Is it hydroxyproline?
Yeah, although we should say, so the remainder of the positions are often occupied, well, many of the remaining positions are occupied by proline and hydroxyproline. So not just hydroxyproline, proline too. Not every single position. There's other things, but many of these remaining positions would be occupied by proline or hydroxyproline. I drew you a specific example of a primary structure in the tutoring notes last time to show what that looks like. Okay, so many of the remaining positions are occupied by proline and hydroxyproline. So now, what type of molecule is proline?
Non-polar.
Yeah, what other group does it go in? What type of molecule is it?
Aliphatic.
Good. Any other categories?
Rigid.
Yeah, I guess it's kind of a vague question. What I was going for is it's an amino acid.
Oh, okay.
I guess it's kind of a vague question, but it's in the group of amino acids. What type of molecule is hydroxyproline?
It's the unusual amino acid.
Yeah, it's also an amino acid. It's not one of the 20 common amino acids that you've memorized. It's not one of the 20 common amino acids you've memorized, but it's still an amino acid. What's the difference between the structure of proline and hydroxyproline?
Hydroxyproline has a hydroxyl group on it.
Where?
On a proline. It's got it on the ring.
Yeah, on the ring. What's the name of this amino acid?
Proline.
What's the name of this amino acid?
Hydroxyproline.
And by the way, the abbreviation for hydroxyproline is HYP. That might be a good thing to memorize. You can see it's got why it's called that. Okay, so something that we need to understand then is, so collagen, in order to have well-formed, strong collagen, you need to have hydroxyproline. Now, the body needs to make hydroxyproline. What do you think the body makes hydroxyproline out of?
An alcohol and a proline.
Okay, I don't know if it I don't know if it uses an alcohol or not, but it uses proline. So the body takes proline and turns it into hydroxyproline by adding an OH group. Now, as you'll be studying later in the course, every reaction in the body requires an enzyme. Every reaction in the body requires an enzyme. So there's an enzyme that's going to catalyze this reaction. Do you know what, we haven't studied enzymes yet, but do you know what does the name of an enzyme usually end with? You might not remember this. Do you remember from other courses what's usually the suffix on an enzyme?
ASE.
You got it. So this is the name of the enzyme that catalyzes this reaction.
Prolyl hydroxylase?
Prolyl hydroxylase. Prolyl hydroxylase. So, what type of molecule is this?
Amino acid.
That's correct. What type of molecule is this?
It's also an amino acid.
Yeah, a less common amino acid. What type of molecule is this?
That's an enzyme.
Excellent.
It's a protein.
Those are both right. It's an enzyme, which is a type of protein. Excellent. How can you tell that from the name? From the ASE. Why is this a logical name for this enzyme?
Because it ends in ace.
Good. But why is the rest of the name logical?
Because it has the PROL, which begins in proline, and the hydroxylase, which is turns it into a hydroxy.
Right. So what do you think this enzyme does, basically?
Adds a hydroxy group to the proline.
So do you see how that makes the name logical? This is called prolyl hydroxylase because it hydroxylates a proline. So oftentimes enzyme names are quite logical. This is hydroxylating a proline, so that's a logical name. All right, now let's see, how much detail do we need to go into here? So many enzymes can have both active and inactive forms. Many enzymes can have both active and inactive forms. Prolyl hydroxylase has an active and an inactive form. In order to keep it in the active form or to change it into the active form, it needs, you need vitamin C. Okay, so what do you think? Suppose you had a test question. What would happen to a person who doesn't eat enough vitamin C? How would you answer a test question? What would happen to someone who doesn't eat enough vitamin C?
They would not have rigidity in their cells. I guess they wouldn't have.
Exactly. So you'd want to go step by step through the logic if that was
Right.
a test question. So what would be the step-by-step logic you would discuss if that was a test question?
If the enzyme requires vitamin C to have the active form hydroxyproline, and someone doesn't have enough vitamin C, then they can't have hydroxyproline. When they don't have hydroxyproline, then the alpha-keratin, or what's it called? The collagen.
Yeah, not alpha-keratin, but collagen.
Right, the collagen cannot exist because it needs hydroxyproline to stabilize the structure of the triple helix.
Okay, so low vitamin C, let's lay out the logic, would lead to what?
Low vitamin C would lead to low hydroxyproline.
Now, that's
low.
What's the intermediate step there?
Low prolyl hydroxylase.
Right, or maybe it's better to say inactive. It'll still exist, but it won't be in its active form. Inactive prolyl hydroxylase. And what goes wrong when you have inactive prolyl hydroxylase?
You don't have enough hydroxyproline.
Low hydroxyproline levels. And then what would go wrong?
Hold on, let me catch up.
Sure, take your time.
Then you would have low collagen levels.
Good. Now, the way your textbook describes it is you would have less stable collagen. So apparently the body would still make its best effort at constructing the collagen, but the collagen would not perform its structural role as well as it did before. So we form less stable collagen. Do you know what the name of the disease is that you get then? That's right. All right. Most notable are bleeding gums, loss of teeth, and periodontal infections. So it doesn't sound very pleasant. All right. So so this is an unpleasant disease that you get from not eating enough vitamin C, especially gum problems. Okay. So this is some logic. It seems from the notes you sent me, it looks like your instructor spent some time talking about this, so I think there's a good chance this will be on the exam. In fact, I don't know how much detail. We could go into more detail. There's a little more detail about this in the textbook, but maybe this is enough for the time being. All right, so here's some of the basic ideas. Notice how you want to try to describe each step of the logic. So, like I said, we could go into even more detail, but to save time, hopefully this will be enough detail for a good answer on the test. So you should try to keep reviewing this so this makes sense to you here about the collagen. Okay, good. By the way, do you know what types of foods you might need to eat more of that are a good source of vitamin C?
Oranges, citrus fruits.
Citrus, that's right. So I think historically, one place this came up is when back when Europeans started taking really long ocean voyages during the age of exploration, when Europeans were trying to sail to India or the Indies or reaching the New World, they'd be out at sea for months and months, and they didn't bring any citrus fruit with them. You know, no one knew about vitamins then. So when they started taking those very long sea voyages, by the time they got back, 3/4 of the crew would be dead from scurvy. And I think it took a couple hundred years before someone figured out that eating more citrus would help. So that's kind of the historical background for this. So around 1500s, scurvy started being a big problem in ocean voyages. And I don't think people have figured out that you needed to eat more citrus until around 1800. So there's about 300 years of pretty bad scurvy. And then I think it was about 100 more years before people discovered vitamins, so people knew why the citrus helped. At first, people just knew that citrus helped prevent it without knowing about vitamins or vitamin C. Okay, so there we go. Let's see. Right, so the point that your instructor made in class was collagen with hydroxyproline is much stronger than without it. So if you don't have hydroxyproline, you get less stable, weaker collagen. Okay, what are the types of secondary structure?
Alpha helix and beta-pleated sheet.
That's right. Remember that the collagen is kind of a digression. It's we just mentioned it. We just talked about it because it's another type of helix, but it's not an alpha helix. So now let's talk about the beta-pleated sheet. What's the slogan for the atoms on the main chain?
Nitrogen, nitrogen, alpha carbon, carboxycarbon.
Let's make sure I got that right. Nitrogen, alpha carbon, carboxycarbon. 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 Do you understand why the nitrogens are on the right, on the lower part, and on the left, on the upper part?
Mm-hmm.
It's just because the strand turned around. It's still the same pattern in both places. Okay, so this is kind of the basic structure here of a beta-pleated sheet. Here's one sheet. This part is not part of the sheet. And here's another sheet. And here's some interactions that can hold that sheet in place. Ha, all right. It takes, you have to draw a lot to get the interactions. So based on what I've drawn here, what types of interactions stabilize the beta-pleated sheet?
Hydrogen bond interactions.
Excellent. Between main chain or side chain?
Between main chain.
Okay. Why is this hydrogen capable of hydrogen bonding?
Because it's attached, or it's got a delta positive charge.
That's right. Why is the oxygen capable of hydrogen bonding?
It has a delta negative charge.
Okay. Is this the same or different from alpha helixes? So the beta-pleated sheet is stabilized by hydrogen bonds between main chain atoms. Is that the same or different from what stabilizes an alpha helix?
It's different because alpha helix includes this and other weak interactions. Let's
see. No, I think that's mistaken. I think that's mistaken. This is getting tough because we've gone over so much stuff. You might easily start to confuse those. But no, the alpha helix, all secondary structures are maintained by hydrogen bonds between main chain elements, including the alpha helix. The alpha helix, well, I think what you were thinking of is alpha-keratin. In alpha-keratin, the two alpha helixes are held to each other by various weak interactions. Do you see the distinction there? So in alpha-keratin, here would be one alpha helix. So here's an alpha helix. What is stabilizing this alpha helix? What's stabilizing the alpha helix is hydrogen bonds between the main chain atoms. Hydrogen bonds between the main chain atoms. Is that the same or different from the beta-pleated sheet?
The same.
But remember, in alpha-keratin, it's pretty hard to draw, but then there would be another, I can't really draw it, but then there would be another alpha helix coiled around the first one. Now, what's holding these two strands to each other? That's what you were thinking of as the various weak interactions. The two coils are held together by various weak interactions and sometimes disulfide links. Tell me when you have those things down.
Okay.
All right, so things are getting serious now. So what holds the shape of a single alpha helix?
Hydrogen bonds
Between
main chain
right.
atoms.
And what holds the two alpha helixes coiled around each other in alpha-keratin?
The various weak interactions.
And
sometimes disulfide links.
Right. And what stabilizes the shape of a beta-pleated sheet?
By the main chain hydrogen bonds.
Okay, so things are getting complicated. It would be easy to get all those ideas confused, so I hope that you're trying to take some pretty clear notes on that. Okay. So here we are back again here with the beta-pleated sheet. So here's the two beta-pleated, here's an example of the beta-pleated sheets, or two strands that go up to make a beta-pleated sheet here. Now, beta-pleated sheets, notice you need to have at least two strands here. Beta-pleated sheets, the two strands could be either parallel or anti-parallel. In beta-pleated sheets, the strands can be either parallel or anti-parallel. How would you classify these strands? Would you classify these strands as parallel or anti-parallel?
Anti-parallel.
Because you can see they turned around here. Notice that's why nitrogen alpha-carbon carboxy-carbon was left to right on the top, but nitrogen alpha-carbon carboxy-carbon was right to left on the bottom. It's possible to arrange these in parallel as well. It's possible for the strands to be either parallel or anti-parallel. There's pictures in your book of the parallel arrangement. All right. So here's a picture of a two beta-pleated sheets.
Right, and the side chains are the ones that are the molecules hanging off of it, right?
That's an excellent point. I should have mentioned that. So the side chains point out to the sides. That's a good point. Do you see these white dots?
It's the hydrogen bonds.
That's right. So here's two. I can't see this in enough detail to see if these are parallel or anti-parallel, but this gives you a better sense of what the beta-pleated sheets look like. It's hard to draw that on the program, but this is kind of what the beta-pleated sheets look like. And as you said, the side chains are pointing off to the side. There's probably a better picture of this in your textbook. What type of structure is this?
Alpha helix.
And what are these white dots?
Hydrogen bonds.
Between what?
Between the main chain atoms.
And what are these white dots?
The hydrogen bonds between the main chain atoms.
They're both secondary structures, so they're both maintained by hydrogen bonds between main chain elements. That's right. Okay. Would you say the alpha helix is relatively condensed or extended in length?
Condensed.
How about the beta-pleated sheet?
Extended.
Do you see the difference? Because if you form a coil, it's not as long as it could be. If you pulled on the two ends, do you see that if you pulled on the two ends of the alpha helix, it would straighten out and be longer?
Yeah.
But notice the beta-pleated sheet is already pulled out, if you see what I'm saying, because the beta-pleated... So that's an important point that I think your instructor emphasized in class. Coiling means, does the alpha helix is shorter, coiling shortens the length. Beta-pleated sheet is extended, so that should be in your notes. The coiling of the alpha helix makes it shorter than if it was extended. The beta-pleated sheet is extended. Okay.
The coil of the alpha helix, say again?
Because the alpha helix is coiled, it's not fully extended.
Mm-hmm.
And because the beta-pleated sheet is not coiled, or just the structure of the beta-pleated sheet is much more fully extended, near fully extended. So what your instructor said you needed to know is know that the beta-pleated sheet is more extended than the alpha helix, oftentimes nearly fully extended.
Okay.
Okay. Can you think of a biological structure that has whose structure is almost all alpha helixes?
A biological DNA.
No,
Oh,
that's a mistake because what type of molecule is DNA?
nucleic acid.
That's right. What type of molecule are alpha helixes?
Proteins.
So do you see how that's a category mistake?
Yeah.
Why did you say DNA? Because you thought they were the same because they both have the word helix in their name. So yeah, DNA is a helix, but it's not an alpha helix. DNA is a helix, just like collagen is a helix, but it's not an alpha helix. Not all helixes are alpha helixes. I wanted to just review something we talked about earlier today. So I was talking about alpha-keratin. What's a structure that's made out of alpha-keratin?
Alpha-keratin is nails and hair.
Hair, or say wool. So your instructor used an example, say, of a wool sweater. What's the structure? What's alpha-keratin made out of?
Alpha-keratin is made out of two coiled alpha helixes.
And it turns out that silk is made out of beta-pleated sheets. Now, most proteins have many different secondary structures and many different parts to them, but silk is made out of a compound that's almost completely or mainly beta-pleated sheets. So silk is not made out of the same thing as wool. So what's the main secondary structure in wool?
Alpha helix.
What's the main secondary structure in silk?
Beta-pleated sheet.
Okay. So your instructor's notes said, be able to explain why wool is stretchy and silk is less stretchy. We need to be able to explain why wool is stretchier than silk. So how would you answer that on the test? Why is wool stretchier than silk?
Because wool is not fully like the alpha-keratin in the wool is not fully extended. Meanwhile, in silk, it's made out of beta-pleated sheets, and beta-pleated sheets are already fully extended.
Good. Talk about that. So beta-pleated sheets are already near fully extended. What did you say about the wool again?
The alpha-keratin is not fully extended.
How do you know? Why not?
Structure because it's an alpha helix.
That's right. And alpha helix is a coil by definition is not fully extended. Because what would happen to a coil if you pulled on the two ends? It would pull into a straight line that was longer, right? So here's the coil. If you pulled on the two ends, it would look like this. It wouldn't be a coil anymore, but it would be longer. So clearly a coil has room to stretch. A coil has room to stretch. Presumably, when you pull on wool, you don't pull out all the coils completely because then it would completely lose its structure. But the point is it can become less coiled as you pull on it. Okay. So it looks like that's an idea that your instructor emphasized that might be on the test, this difference between how wool and silk behave. One thing I wanted to mention is remember that beta-pleated sheets have to have at least 2 strands that are hydrogen bonded like this. But oftentimes they have many strands. So I didn't write the whole thing out, but oftentimes, I'm running out of room here, but oftentimes there would be another strand, then another strand, then another strand. Oftentimes there would be 4, 5, or 6 strands all hydrogen bonded to each other in the beta-pleated sheet. And that extensive hydrogen bonding can lend a lot of strength to the substance. For example, that extensive hydrogen bonded structure could lend a lot of strength, say, to silk. So silk gets a lot of its strength by the extensive hydrogen bonding in the structure. Remember, there could be multiple strands all hydrogen bonded to each other. Okay, I think that covers the key points that your instructor made about secondary structure. So we can move on we can move on from that. But it's things are getting a little complicated now, so it's important to study that. So what I've been trying to do is I've been trying to use the typed notes that you sent me. I'm just trying to focus on the ideas that your instructors seem to focus on in class. Okay, so we went over today these key types of secondary structure. So this takes a lot of work to keep this all well memorized. There's really a lot of memorization in biochemistry. There's a lot of memorization, so no time like the present to memorize this. So try to make sure that you can answer test questions about, say, hair, wool, teeth, cartilage, silk. Silk is the new one. What's silk made of?
Beta-pleated sheets.
Right. So that's a new concept we hadn't talked about before. So we talked about alpha-keratin, beta-pleated sheets, and collagen. Those are the main types of structures we've been focusing on the last couple of sessions. So you should carefully review these notes and the different levels of structure that we have. We talked about disulfide linkages. Remember, the place we've seen disulfide linkages play a role so far is holding together the coiled coils of alpha-keratin. So that's something that could come up. Remember, we saw that's why nails are more rigid than hair, because they have more disulfide linkages. Here's the structure of collagen. We talked about how the body has to make hydroxyproline, and the enzyme that it uses to make that requires vitamin C to stay in the active form. I think there's a real good chance that'll be on the test. Okay, now one thing to keep in mind is every single thing I talked to you about today, you already heard about in lecture. I'm just covering the stuff from lecture. So every single thing we talked about today, you already heard about in lecture, but most of this stuff I don't think you were remembering from lecture, right? Now, the moral of that is most of this stuff you won't remember from the tutoring either, unless you make a really strong effort to put in a lot of time reviewing this session. You might want to rewatch the video or at least go through the notes. Remember, just going through the material once is not enough because you already did that in lecture and it didn't stick, right? So you're not going to learn this material just from having gone over it in the tutoring. It's important to make a serious time investment to try to have very clear notes about all the various different levels and structures here. Because obviously the next time we meet, we want to move on to some new material. But that requires a serious time investment on your part to review this. Because like I said, you can tell that just going over it once is not enough, or you would have already learned it from the class lecture. So just going over it in the tutoring is not enough. You need to put in some serious time reviewing that. But I hope that your notes are clear so that you'll be able to work on that.